Hello everyone, thank you very much for joining this Barhem webinar on peptide aggregation, a complex topic. Before jumping into the presentation, let me introduce our speakers for today. The first part of this webinar will be presented by Patrick Plattner, Senior Group Leader in Quality Control at Barhem, and the second part will be presented by Hova Siu, Scientist in Patrick's group. I will be your moderator for today's webinar, and I'm a Product Manager at Barhem. Before starting, I would like to add a disclosure. This webinar that you will see is pre-recorded, however, the Q&A session afterwards will be live. With this being said, let me quickly introduce Barhem. We are the leading CDMO for the development and manufacturing of peptides and oligonucleotides, API. With more than 50 years of experience, our success is based on long-term partnerships with pharma and biotech companies. With them, we contribute to bringing new medicines on the market and helping patients worldwide. As a company, our success is based on 5 strategic pillars: people and culture, innovation and technology, sustainability, customer centricity and service, and the last one, operational excellence and quality. We are growing really fast and we set up ambitious goals for the future, and to support this growth and ambition, we expand our capacity and capability for manufacturing. After a capital increase in 2021, we installed additional capacities and started the construction of a new tides manufacturing building, which is close to be operative and will be the most modern facility for large scale. And this year we have announced the acquisition of a new site in Switzerland, which will be ready by the end of the decade. These investments are also a proof of our commitment to peptide and oligonucleotide science, which you will see a very nice example later on this webinar. With no further ado, I leave the stage to you, Patrick.
Thank you, Elodie, for the kind introduction into the company and ourselves. Welcome to the audience. My colleague Hova and me are going to talk about aggregation. Aggregation is an interesting, wide topic with lots of research going on using plenty of analytics. Our focus today will be the perspective of a peptide manufacturer. Peptides are our daily challenge at Barhem. Consisting of amino acids, they are a small version of proteins, chemically synthesized. This indeed generates a further category of drug products between small molecules and biologics. For a long time, there was only very few guidance from the authorities for peptides, which changed in the last year and was triggered when chemical synthesis became prior over recombinant production for medium-sized peptides like glucagon. And if we are talking about glucagon, there we are. It is widely known to aggregate, so peptides can do aggregate, and therefore we have a serious issue. Aggregates are a consequence of physical instability. But what are the concerns about? The authorities, FDA and EMA, give guidance for biotherapeutics to address the impact of immunogenicity, and therein the role of aggregates is addressed due to efficiency and safety reasons. Imagine a product forms aggregates, then drug substance is removed from the drug product and the effectiveness of the healing success is questioned. From safety aspects, the worst case is an immune response, triggered by aggregates. The reaction of human body can lead from a neutralizing clearing effect of the peptide up to acute reactions like an anaphylactic shock. The last issue is more related to practical considerations, but very important to us. As a manufacturer, you want to have a smooth and projectable synthesis without clogging, geeling, aggregation, or other physical instabilities. The currently most promising drug product entering the market are peptides based on GLP-1 or GIP, addressing diabetes-related diseases. Handling many of them in-house let us learn a lot about aggregation behavior. There's also plenty of literature about aggregation that attempts to increase the physical stability, of course without losing the activity. Small changes, like between liraglutide and semaglutide, can change many peptide properties, also the aggregation behavior. Therefore, the aggregation behavior is specific and must be assessed and managed individually for each peptide. Let's look a bit inside the mechanism and the criticality of the different aggregation phases. In the beginning, we have the so-called lag phase. Everything is fine, maybe there are different secondary structures or higher molecular aggregates, but all reversible. It turns into a problem if this changes and it becomes energetically more favored that higher order structures are growing, fed by direct nucleation from the monomer. This might even be speeded up by fragmentation of the aggregates, respectively fibrils, causing increasing amounts of nucleation sites. Now we are in acute trouble. In between, there's an initiation, an initial nucleation process. Those aggregates are called seeds, and if they are present, they can start an aggregation process, even at a later time point, somewhere in the future. And therefore, they are relevant for the aggregation propensity of the final material. Hence, to avoid seeds and acute problems, the synthesis must be optimized already towards the initiation of aggregation. Here, the whole manufacturing process is shown, from the SPPS on the left to the final white powder coming from the freeze dryer on the right. To manage aggregation is mostly a topic of the downstream. They are the purification steps allowing a depletion of higher molecular weight species, together with the chemical impurities. At the same time, there is a risk to generate new seeds or to run into solubility/aggregation issues. If we switch from the production step to the unfavored stress factors applied there, we find a lot of mechanical influences coming from surfaces. Of course, they are the materials of the equipment, then air, solution, or even solution ice surfaces, as we can have them during freeze drying. On the other hand, we have the solution characteristics, as they are the buffer type and strengths. We have organic solvents in our systems, pH and pH shifts take place, temperature and duration is important for holding solutions, and of course the peptide concentration itself plays a role. But what can we do? We need to avoid inappropriate solution conditions and find and work in the best possible operation window our solutions can offer. And to define those, we need analytical support. Now I forward to Hova, who gives us an insight into aggregation analytics, advantages and relevant handling aspects of selected techniques.
Thank you, Patrick. How do we analyze peptide aggregation in Bachem? We have a plethora of analytical techniques in-house to characterize peptides and in particular the aggregation propensities. Those analytical techniques can be divided into different groups, such as for the characterization of soluble aggregates, for the analysis of subvisible and visible particles, and we can also access protein folding states, since many aggregates are formed of secondary structural elements such as beta sheets. Many analytical techniques can lead to a variety of results and many discussions. Therefore, it's highly desirable to have a few selected methods which can address aggregation propensities of peptides. The presented method here can cover a broad size scale, starting from the initial stage of aggregation, from a monomer to an oligomer over larger aggregates in the visible area. Soluble aggregates can be studied with size exclusion chromatography. In the size exclusion chromatography, the size range is dependent on the pore size of the SEC column and the analyzed species. The pore size should be 2 to 3 times larger than the biggest analyzed molecule. An UHPLC is used, which provides many advantages, such as less solvent consumption and highest resolution. The separation is based on the hydrodynamic radii of the molecules. In this picture, you can see larger particles in green, which elutes faster through the column in comparison to the smaller blue particles. The blue particles are assigned to the monomeric peak in the chromatogram below, and the dimer is assigned to the larger green particles. The hydrodynamic radius is dependent on the molecule size, the degree of folding, and the particle shape. A folded peptide has a smaller hydrodynamic radius in comparison to the unfolded one. It's possible to specify and quantify the oligomeric species as shown for the dimer. It's possible to separate oligomers to aggregates from 1 to 1000 kilodalton dependent on the column pore size. SEC provides many advantages. It's quantitative, robust, and sensitive with an LOQ of 0.1%. We can use it in a high throughput manner and inject sample one after each other. It is possible to characterize the oligomeric species by coupling a multi-angle light scattering detector, which gives information about the molecular weight of the species. Of course, SEC has also its limitations. It needs time for method development, and the sample preparation can already impact the high molecular weight content. Therefore, it's important to have a suitable solvent where the peptide is stable over the analysis time. Also, a chromatographic condition can change the high molecular weight content composition. We cannot detect any larger aggregates which are not soluble. We cannot perform measurement in parallel. It's also not possible to differentiate between the different oligomeric species, which might be critical for aggregation later on in the final material. Another method to characterize visible particles is based on turbidimetry and visual inspection. Here we can screen multiple variables at the same time. In principle, you track the aggregate formation by use of turbidimetry or visual inspection. In the picture, you can see 3 different types of solution. Above, you see a turbid solution. Below, you see an example for particle formation, and next to it, an example for gelling. Disadvantage of this method is it is very insensitive. So there are many advantages since we don't need any time for method development. We have a huge amount of sample volume, which allows us to use it for other analytics to characterize the high molecular weight content and also to quantify them. So as you can see, you can detect different types of aggregates, and it's possible to apply different stress conditions at the same time. This method allows us a systematic screening of different stress conditions. So the variables which can be screened are pH, product concentration, and temperature. If you collect all this data, you can generate such a contour plot to find the robust operating window for your peptide in solution. This is shown here in the contour plot, where we have the blue area for the robust operating window at the lowest temperature. If we increase the temperature, you can see that the aggregate level is increasing. So pH and temperature impacts the aggregation behavior of your peptide in solution. Another nice method to detect larger aggregates is based on dynamic light scattering. So here we can also monitor the initial steps of oligomer formation and also larger aggregates in one technique. How does dynamic light scattering work? In this scheme, you can see that the red laser beam is hit our particle in solution, and the scattered light is recorded by a photodiode which monitors the intensity over time. Larger particles diffuse slower relative to each other in comparison to smaller particles. So you see a faster change from the intensity for the smaller particles. In the autocorrelation analysis, you can monitor the decay of the larger particles, which are slower in comparison to the smaller particles, and the decay rate is proportional to the diffusion decay, and from the diffusion coefficient you can recalculate the hydrodynamic radius. The hydrodynamic radius is dependent on the molecular weight, the folding, and the particle shape. The intensity scales with the hydrodynamic radius by the power of 6. This means that you can also detect a small amount of larger particles, which is not critical for quality control. Furthermore, we can also access multi-component system. This is nicely shown in this autocorrelation function. This bimodal curve indicates the presence of smaller species and larger species. This is also shown in the corresponding histogram, where you can see larger particles with larger hydrodynamic radius and smaller particles with smaller hydrodynamic radius. So dynamic light scattering provide many advantages. It's generic, we don't need any method development, and it's possible to apply a lot of sample on such a 384 well plate. So we can establish measurements in parallel with positive controls, negative controls, and reference substances as triplicates. It's very well suited for comparison study of physically unstable samples in comparison to size exclusion chromatography, where the sample have to be injected one after each other, and during analysis time you can already have degradation of your peptide. So we can generate quite a lot of data with 384 well plate. Thus it's very important to pre-evaluate these data to find reliable results and exclude bad results. In this experiment here, we use different types of filters, filter 1, 2, and 3, and an experiment without filter. Below you see that many data points are already excluded without filter, and even more data points are excluded with filter 1. This indicates that the peptide interacts with the filter 1 and leads to a low data quality. Filter 2 and filter 3 have comparable good results. Another well-established method is based on THT assay. Here we can screen the formation of better structured aggregates. In principle, a fluorescence dye is used which can be excited at 349 nanometers. It has a free rotation around the axis. Upon binding to a better structured fibril, it cannot rotate anymore, and it emits a fluorescence band at around 454 nanometers, and you can nicely monitor the increase of fluorescence intensity over time upon binding of the fluorescence dye to the fibril. This technique has many advantages. It's already well established and it's more sensitive. It's generic. We don't need any method development. And you can apply it for peptides without any intrinsic fluorescence. Limitations is that THD only bind to specific particles with a better structure. That means it can lead to false positive results in case if you have a beta sheet in a monomeric state, and you can also have negative results for amorphous aggregates which does not have any structure. So such an experiment takes about 1 week. Here we can again see the kinetics of fibril formation, and it takes a long time. Thus you can see we have got long lag phases, and during the lag phase we have a high variability of our system. You can reduce the analysis time by applying a stress condition, for example temperature or steering, and then you also reduce your lag phase. In the last example you see you can also have a one-time measurement where we apply our measurement after application of a stress condition. This reduces the measurement time to a suitable analysis time. To sum up, we have learned about the aggregation propensity of peptides during the manufacturing process and different analytics to monitor smaller and larger aggregates. It's very important to know about the physical stability of your peptide. Every peptide has its own trigger for aggregation. It's peptide specific. And already standard manufacturing processes can lead to aggregation of peptides. That's why we have to set individual criteria with respect to safety, shelf life, process, and analytical capabilities. Another important point is to find the optimal operating window. So we have to optimize the process condition of the downstream process to deplete any kind of high molecular weight species which can induce aggregation in our final material. Furthermore, seeds which can induce in the final material has to be avoided to control our material. We believe that a profound expertise on the applied analytical methods is highly important to generate high quality data. Up to date, there is no single technique applicable to cover all analytical challenges. We are convinced that a few selected method is highly sufficient to address aggregation propensities of peptides. With this, I want to hand over to Elodie for the poll time.
Hello and
welcome to everyone. We are now live. So before starting the live Q&A session, we would like to ask you a poll question. Have you experienced already aggregation issues with the peptides you are working on? So we are waiting just a bit, the time that the audience answer the question. And during this time, I would like to thank Hova and Patrick for this nice overview on the challenges of peptide aggregation and also the solution for the analytical solution methods. So we have now the result of the poll question. So it seems that, yeah, a majority of you had issues with this aggregation, so almost 70%. And the rest of the audience did not have issue, but mainly because they did not work with peptides, or it was really easy peptide to handle. So thank you. Now we will start the Q&A session. And we have some question of the audience. I think the first one would be for Patrick. Can you share an example on the consequences of detecting a molecule's aggregates? And then there is a second part of the question, so maybe you can already answer this part.
Yeah, thank you, Elodie. Thank you for the question. Yeah, we, of course, observe aggregates during our processes. This can be either during filtration processes or other manufacturing steps, and then we try to optimize it. And with our process, we have a lot of opportunities. For example, during process development of liraglutide, we had smaller aggregates, which then lead at that point to an additional chromatography we implemented in order to remove all these aggregates prior to the continuing the process. And with this, of course, analytic is required to find the way to the better procedure and to determine whether the improvements we made are really valuable.
Thank you, Patrick. Actually, another question that is related to this one is how to quantify the fibrils of this aggregate. Maybe Hova then.
Yes, with the THT assay, actually, you can monitor the formation of fibrils. And if you have reference substances, then of course you can have like a calibration, and then in the end, you have like a kinetic of your fibril, and then you can measure or see the fluorescence intensity of the plateau which is forming, because at the beginning you have your monomeric species, and then you have your lag phase, and then you have the slope, where in the end you have your fibril, and then you can try to quantify this via the fluorescence intensity.
Thank you. Another one regarding THT. So you mentioned that the THT assay takes 1 week. Does that indicate that you stress the samples temperature shaking for up to 1 week to obtain the results and answers?
Yes, this is the normal procedure. If you unstress your sample, then it of course takes time until oligomerization takes place. So you have your monomeric structure, and then it depends on the peptide sequence if it's prone to aggregation or not. And you can reduce the time of the lag phase by applying stress conditions such as temperature, or you can use mechanical stressing and reduce this to a suitable analysis time that maybe takes only 2 hours.
Okay, thank you, Hova, for this clarification. Then more regulatory question. Do aggregates need to be monitored during stability studies? I guess Patrick can answer that.
I think this is more applicable to drug product analytics. So since we're doing drug substances, the regulatory aspect is not as high as for the drug product. Of course, we have an interest in the stability of our peptides in the drug product itself, regarding the aggregation propensity there, which can come from our material. But if everything went fine, we have just peptide there which are still in the area of being dynamically and adapting themselves to the solution conditions, and those will be defined during in the drug product formulation.
Okay. Thank you, Patrick. Another question, do you have experience with aggregation index analytical methods? And would you recommend it for characterization purposes?
Honestly, no. Aggregation index analytics doesn't mean something to me, but it sounds interesting. I definitely will have a look at it.
Good, thank you. So, okay, we have plenty of questions regarding like more technical aspects. So for, I guess this one will be for Hova. For the SEC, so you mentioned that the sample preparation may impact the aggregates, inducing or reducing it. Could dilution of a sample solution before injecting it in the system have an impact?
Yes, if you dilute your sample, of course, you reduce the concentration, and that might lead to the dissolving of the aggregates. But in case if it's already a matured aggregate, then of course it's not possible. First, you have to screen, let's say, different variables that you have the right composition of your solvent, which allows your peptide to be stable during the analysis time. So it's good to screen variables such as pH, concentration, azethonitrile concentration, or the salt, to find a robust operating window.
Okay, thank you. Now a bit more regarding the process, and I guess more for Patrick. Is it challenging to control aggregation during lyophilization? And is the rate concentration dependent?
So there's literature showing that the freeze drying process has an influence on there can aggregation or aggregates develop, which later causing aggregation or increased aggregation propensity, and that's the point where we have to check our materials. So for the propensity, we need to perform kinetic assays to see how fast it's developing compared to material where we know the aggregation propensity. Of course, seeds as such you can determine by SEC, but either they are stable or, as Hova said before, can be changed in the quantity during the chromatographic analysis during dissolution or during the elution time.
Thank you. Now there is questions regarding actually the GLP-1 type peptides that are, I mean, the new ones that are with lipids on their chain. Is this lipidation of the peptide affect the aggregation? And also, how is it possible to differentiate between the covalent aggregation or non-covalent aggregation?
Yeah, the covalent aggregation can be tested, for example, with SDS page. So you dissolve everything and see whether you still have the aggregates in. With peptides, you quite often have covalent aggregates, especially if you have disulfide bridges, but it can be caused also in other ways, and usually there you don't have such big problems with the SEC because they are stable and they you can run them with a high organic content to measure them.
Thank you, Patrick. Then I guess a question more for Hova now. You mentioned with the THT assay that you can monitor the beta structure aggregates. Now, how can we determine, sorry, the alpha helix structure if there are?
Oh, thanks a lot for this question. So THT is actually specific to beta structured aggregates. So it has to be like having extended beta sheets. And if you want to characterize alpha helical structure or other ones, then a way to go is using, for example, vibrational spectroscopy. You can use FTIR spectroscopy, where you can look at the polypeptide backbone chain, and you have the carbonyl vibration, and it gives a specific signature in the amide 1 region, where you really can look at monomeric beta sheets at 1630 wave numbers. And in case of an extended beta sheets, for example, it would be shifted to lower wave frequencies like 1615, and then it's possible actually also to differentiate between monomeric and extended beta sheets, and it also allows to characterize alpha helical structures. Normally it's found between 1640 and 1615 wave numbers as signature, but also CD spectroscopy is a really powerful tool to look at secondary structures, in particular for alpha helical peptides, where you have got, let's say, the ellipticity, where you have got two negative bands at 212 nanometers and 220 nanometers, which gives indication of an alpha helical structure. And here you can also see conformational changes over time. And if so, then you will have also specific signature for the beta sheet.
Thank you, Hova, for this answer. Then for Patrick, during your process, how can you identify that your peptide is aggregating, and what would be, what is a typical time scale for aggregation? And then the last part of this question would be if it's possible to implement IPC or in-process control like J-pont during your manufacturing process to control that.
Yeah, often it starts that we have an issue. So for some peptides, especially if you see that they're based on GLPs, for example, you can expect to get into trouble, and then very early you can start evaluate the potential aggregation issues. But in fact, quite often you're starting a project very small, beginning with the synthesis, and you start to collect information. And that's very important to have this in mind and to look for indications you might get a problem later, especially if you have high back pressure during filtration steps or something like this. So these are early indications then, and if you notice those, it's good to have a closer look to it and to stress the material under the conditions you're applying in the manufacturing process. So usually you have a range of organic solvents and buffer systems you can use. The focus is on the purification, and mostly this is for the chemical impurities. But of course, in a certain range, you can also adapt then this process into the direction to look more for the physical stability, which is not only the aggregation as such. It can be any kind of solution problems which can be solved then going that route. And of course, we do this performing analytic in parallel. We're testing at least our THT and DLS as in first instance. If you have solution, DLS has a high advantage because we are very generic there, and then we can measure samples in parallel to direct the process development into the right direction.
Thank you. And just to continue on this like process aspect, can you see a difference of the aggregation propensity between different techniques during the lyophilization, for instance, if it's spray drying or I mean, during the isolation of the peptide, if it's spray drying or lyophilization?
I suppose if it differs, we would see it, but honestly, we have very few products where spray drying is used, and these products do not tend to aggregate. So my own experience is very low to answer this question.
Thank you. Okay, so our audience is also interesting in knowing also if it's services that we do offer in our I mean for our customers. Is it something that
within a project, yes, we are doing it, and we also try to go into the direction if we see that we get problems during the manufacturing process, but not independent of any project. We are a peptide manufacturer, so no analytical services. In certain cases, if the customer needs our help, of course, we are open for discussions. Yeah.
Okay, thank you very much for this clarification for our audience. So there is a question regarding actually the specification for a drug products, but yeah, since the aggregation and fibrillation propensity is affecting the quality of the drug product, is there physical, I mean, either specification on the physical property of the API that can be set up prior to that in order to mitigate this risk of aggregation?
We try to optimize our process to the best in order to not run into the problem at all. But of course, we are also in contact with our customers in that point, and it also leaded to the request to have a closer look and to reevaluate our aggregation propensity. Finally, this ended up to have a in-process control and to watch at the critical steps in order to generate aggregation-free or seeds-free material which are suitable for the drug product requirements of our customer.
Thank you, Patrick. Then there is, I guess, a last question for Hova. So beta-structured aggregates can be investigated with a THT assay, but is it also possible to use a complementary method for monitoring this beta structure?
Thanks, Elodie. This was a question I have already answered somehow, but of course you can try to use CD spectroscopy, as I mentioned, or FTIR spectroscopy to look at the secondary structures of those peptides. So you can confirm then actually if it's a better structure from a THT assay, and then use FTIR and CDS complementary methods to confirm that.
And thank you, Hova. Okay, then it seems that our audience has no more questions. We answered a lot. So with this webinar, so you could learn from our experts, Patrick and Hova, the techniques that can benefit your daily work and help to address this challenging that you face with these aggregation issues. So now we'll invite you to visit our website and check our brochures and also previous webinars. I would like to thank you all for attending this webinar and wish you a great rest of your day. Thank you.